Welcome to Life and Biology. This is Dr. Joel Graff, and this is going to be some lecture notes from February 11th lecture for my Principles of Living Systems course. It's a first-year biology course, cell and molecular biology course at Montana Tech. Anyway, we had a quiz to start the day on Friday, and so the lecture is a little bit shorter than usual. Also, making it shorter than usual is the idea that, or the fact that we didn't finish walking through all the different cell structures and cell images on Wednesday's class, and so we took some time finishing that up on Friday. So we only have 2 slides to go through for this lecture. The first slide is where we're going to take prokaryotes, which include 2 branches of life, the bacteria and the archaea, domains of life, and compare it to the third domain of life, which is the eukaryote. And remember, eukaryotes occurred when an archaea ate a bacteria. And then later on, that new eukaryote actually ate some more bacteria, and so then they turned into things like chloroplasts for plants and other plastids in the plant cells. So it wasn't a only one-time thing, but each time was a big event. The first time we had eukaryotes being born, and the second time we had a eukaryotic cell eat a bacteria. That was the beginning of photosynthesis that was the beginning of eukaryotic cells that could carry out photosynthesis. It ate a photosynthetic bacteria. Okay, anyway, so we did some time comparing and contrasting after we looked at all of those images to kind of summarize what we saw. Some similarities between prokaryotes and eukaryotes, that is, similarities for all 3 branches of life, are that they all have a plasma membrane. They all have ribosomes, which is the enzyme for reading an mRNA and translating it into a protein. They all have DNA. They all have some cytoplasm, that's the fluid inside the plasma membrane. They all had membraneless organelles. It's often said that prokaryotes don't have any organelles, and that's not really true. They have different areas of their cell that act essentially as organelles, but they just don't have a membrane that is the surface of them. In the lecture, or in the images that I showed, I talked quite a bit about different membraneless organelles in eukaryotic cells that we're now aware of, including one that is nearly as big as the ER endoplasmic reticulum itself. So these are very important to cell biology, and even though you've probably been learning about different parts of a cell your whole life since elementary school, these are ones that are often ignored, and we're finding new ones all the time. Finally, all cells have flagella. Now, you might not think that human cells have a flagella, although you probably know that a sperm does. But all the cells have flagella. It's just that most of the cells in a human, the flagella is kind of retracted to the point where it's not sticking out of the cell, and it's basically just a shadow of its former self. But anyway, those are some similarities between prokaryotes and eukaryotes. And if we're thinking about the tree of life, we might also remember that we all came from a common ancestor. So if all 3 domains of life have all 3 or have all of these features, we can basically assume that the last universal common ancestor had these different features as well. Okay, so what are some differences between prokaryotes and eukaryotes? Well, cell walls. Prokaryotes, they have cell walls, both bacteria and archaea. Now, eukaryotes, the answer is sometimes. For example, yeast in plant cells have cell walls, but many other branches of the eukaryotic tree, including animal cells, do not have cell walls. Capsules, this is a sugary coating that you can find on prokaryotes. The prokaryotes have them, but not always. I should have written sometimes here. Excuse me for that mistake. But then eukaryotes, they don't have capsules, but they do have some other stuff on the outside of the cell, and we will talk through some of that stuff later on. And then membrane-bound organelles. Prokaryotes don't have these. They don't even have a nucleus. The nucleus is basically the main feature of a eukaryotic cell that they notice first. If you look at eukaryotic cells through a microscope, you can often see the nucleus even without the help of a stain of any sort. But prokaryotic cells do not have a nucleus or any other membrane organelles. Okay. Then the last slide for that day was talking about cell size. Now, we know that there's some things that can go into and out of a cell based off of just diffusion alone. So that could be nutrients and gases. Here I have oxygen going into the cell and CO2 going out, because that's what an animal cell would be. But sometimes plant cells might have carbon dioxide coming in and oxygen going out. At other times, plant cells might have oxygen coming in and CO2 coming out. It just depends on if they're actively doing photosynthesis or whether they're living off the sugar that they made up. Okay. Anyway, with diffusion, we know that this is a process that can take time. If you heat things up, things go faster. But because of diffusion, there is a maximum size that cells can have. And the limit is for gas exchange and other nutrient exchange. And so that's all based on diffusion. And diffusion's a slow process. If a cell is too big, then parts of the cell don't get enough nutrients or gases and the waste piles up. So what is too big? Well, it's when the volume of a cell is much too large for the amount of surface area of the cell, especially if the cell tends to be kind of a round shape. Humans have some cells that are very long called nerves, but they're very thin, and so then they don't really mess with the surface area to volume ratio too badly. But let's say we have cells that are circles in general, spheres. We could take the ratio, the surface area, the surface of our sphere versus the volume of the sphere, and a small cell will have a large surface area to volume ratio, and large cells have a lower relative surface area to volume. And why is that? Is that the bigger a cell gets, the surface area goes up by a power of 2, because area is a squared measurement, whereas the volume goes up by a power of 3. And so as the cell gets bigger and bigger, the power of 3 will quickly overtake the power of 2, and the ratio will be not consistent as the cell changes shape and size. Okay, so that was a very short discussion covering mostly just these two prokaryotic cells versus eukaryotes, and also why can't cells be huge. Again, that was because we covered a bunch of images of cells from a previous lecture that we hadn't finished. All right, this has been Life in Biology. I'm Dr. Joel Graff, and you can like and subscribe or not.